What's up, man?
Not much, buddy.
Sorry.
It's all good.
Okay.
Hey, good morning.
I was doing the gifted and talented. I totally forgot until like 30 minutes ago.
We are already live, too.
Good.
So,
Thank you.
yep.
Streaming?
Yep.
Here. Go.
Can you at least start it, please?
Wait, hold on. Wait. Come here. Step out, guys. Start working. Remember, Charlie, do you have your headphones? Okay. You got this.
Yeah. Spice it up a little bit in the morning, right?
Is Mr. Ewald still gonna come back?
Yep.
Put allegiance to the flag of the United States of America and to the republic for which it stands, one nation, under God, indivisible, with liberty and justice for all.
Seriously?
Proud to be an innocent, and proud to be here. Proud to be my dad is
Daddy? Daddy,
Yes, honey. How can I help
will
you?
I have to say on my mask right now?
You have yes, because Mr. Ewald will be here.
Wait, so I can take off my mask? Like, I still don't have my mask on.
Honey, I tied it on for you.
I know. That's not too comfortable. Can you tighten it?
That's Jean. Do you have anybody? Oh, we have a lot of people here. All right, I'm gonna go ahead and say something.
Yeah.
Alrighty, guys. Good morning. It's good to see you guys. Metaphorically, we will start here in about 5 minutes. Okay, just get everybody an opportunity to kind of jump in. So, if you need to go take that restroom break, if you need to go get that cup of coffee, maybe, or if you need to go get something to eat, we got 5 minutes. So we'll start at 8:10. All right, so Mr. Ewald is putting this into the chat, but just want to remind you guys, remember that you want to have a sheet of paper out as well as something to write with. You'll be taking your notes. You'll also be answering questions that we're going to have on the presentation. So just make sure to have something out to write on and also to write with.
All right, I'm going to hop on real quick too. Mr. Ewald, welcome everybody. Thank you to Mr. Wen's class, thank you to Mrs. Becker's class, and of course thank you to my class for logging on and joining us for this live lecture. Like Mr. Wen said, we're going to start here in about a minute or two with the actual material. So if you're out there, get comfy. We got some really cool, fun stuff that we're going to be talking about and showing you today, so we're all excited. All right? Get with you here soon. We're good. It's right at 8:10, so.
Alrighty, so how this lecture is going to go today is we're going to talk about some background information associated with the cell membrane, just to make sure and to stress the importance of understanding the foundation behind the cell membrane. Then we're going to talk about new stuff in terms of passive transport and why passive transport is so important and how passive transport actually works. Then we are going to do some demonstrations interspersed all throughout this live lecture, but hopefully you guys enjoy it, and hopefully you guys have, if you have questions, that you want to make sure to ask those in chat. So what we are talking about today is the cell membrane, which is also called the plasma membrane. So that's exactly what it sounds. It's a thin, flexible boundary between the cell and its environment, and its primary role, its primary function. The reason why it's so important is because it regulates the movement of materials in and out of the cell. So we have this cell membrane right here, and you can see that this cell membrane is interspersed with all different types of molecules. So it is a pretty complicated structure, but we're going to try to simplify it as best as we can. In terms of selective permeability, so let's look at those two words separately. Okay? So permeability means the ability for something to pass through something else. So in terms of fluids, fluids can be permeable through a membrane or through a thin film of some variety. So that permeability is the ability for that fluid to go through another substance. Selective means that we can filter out certain things that we don't want. And so with our cell membranes are selectively permeable, which means that it has the ability to control things that move in and also things that move out. So some of the things that we want in, we want things like oxygen. We want carbon dioxide to go out. We want sugar to come in. We want waste to go out. So all different things that we want to make sure to be able to control. Now, another thing as well, guys, I know I talk really fast. My students have told me that already. So if I'm talking too fast, if I'm going too fast and you don't have the ability to copy the notes down in time, let me know that. And that way we can slow down and we want to make sure that everyone is able to get all this information. Now, the plasma membrane is made up of a phospholipid bilayer. So let's break this word down, bilayer. So that prefix bi means two, which means that it has two layers. Our cell membrane is made up of two layers, and those two layers are going to be made up of phospholipids. So what is a phospholipid? It is a molecule that's composed of a polar phosphate head and a nonpolar fatty acid tail. In my class, I described it as the jellyfish-like substance that makes up your cell membrane. Now, from this picture and what we just talked about the polar phosphate head and the fatty acid tails, you can see the outside of the bilayers are made up of the polar phosphate head, and the inside of the bilayer is made up of the nonpolar fatty acid tails. So, why is that important? Well, it's because we want to make sure that we keep this idea of fluid in terms of the behavior of the cell membrane. So the cell membrane behaves like a fluid mosaic model. What that means is that these bilayers will float around and they will adjust to whatever we need in terms of being able to adapt to our environment. So our cell membrane is constantly facing different types of environments, and so it needs to be able to rearrange these molecules to accommodate for those different changes. And so that's exactly what that fluid is. So if our cell membranes were static, they didn't move, that would be very, very bad. We would die very quickly. But instead, our membranes are fluid, which means that it's constantly moving and constantly changing. In terms of the mosaic, so the mosaic is in reference to all these different molecules that you see embedded in and on the cell membrane. So we're going to talk about proteins, and we're going to talk about membrane proteins and channel proteins and how they're important in passive transport. But you can also see in terms of the cell membrane, remember how I mentioned it was complicated. Well, we have all these different molecules here. We have some glycoproteins, we have some cholesterol that's embedded. Those are obviously really important, but today's lesson, we want to make sure that you focus on the membrane proteins. Okay, so passive transport. It's the way in which our particles move across the cell membrane, and I want you to make sure that you star, you underline, you put an asterisk next to these words without using energy. So again, like I said before, our cells need to be able to get different types of nutrients into the cells. And so we want to make sure that our cells are able to survive. Well, one of the ways in which we're able to do that is through passive transport. And so what I want you to think about is I want you to think about an analogy. So you are at the Ohio River. You rent a canoe. Let's not swim in the Ohio River, okay? That's super dangerous. But if you are getting a canoe and you're renting a canoe, you put the canoe in the river, and then you're going downstream. Is it going to require any energy at all for that canoe to go down the river? Probably not, okay? So in that instance, that's an idea of what passive transport is like. Now we have three modes of passive transport that we're going to talk about today. We're going to talk about diffusion, facilitated diffusion, and osmosis. So diffusion is the movement of particles from an area of high concentration to an area of low concentration. Again, this is natural and does not require any energy at all. An example of this is if you have that person that sprays that Axe body spray or that perfume, and then all of a sudden it starts to smell up the room. Absolutely 100% that's going to be diffusion, because it goes from that individual, which has a high concentration, to around the room, which has a lesser concentration. So it's going to spread throughout the room. Okay, I'm going to actually show a very, very quick demonstration of this, but I'm going to need your help first. All right, so I got four colors here. I got blue, yellow, green, and red. Okay, I want you to choose a color in the chat on which one I'm going to use for this demonstration. Okay, so blue, yellow, green, or red.
All right, let's get those answers in the chat. Blue, yellow, red, or green. Which color do you want Mr. Wen to use? Oh, we got some coming in here. Ooh, 3 blues. We got a green, a red. We got another red.
It's a tight battle between blue and red.
It is. Blue and red are kind of in the lead right now. But blue, it looks like, is taking a pretty demanding lead. So I think, based on the responses from our kiddos, we probably need to go with, Highlands and UK blue.
Highlands, UK? No, I'm thinking of Memphis blue.
I don't know about that.
Memphis blue, home of the blues. I think that's what we're really focusing on.
They shouldn't say Duke blue.
That's true. That's true. All right, so here's what we're going to do. So we're going to use this food coloring, and this food coloring is blue. And so you can see that this cup of water, it's just a very simple cup of water, and all we're going to do is, this is our high concentration, okay? That's where most of that blue is coming in. We're going to put it in a container that is of a low concentration. So what happens as a result of that? And you can see that it's starting to spread out. It's starting to diffuse. It's requiring no energy whatsoever in order for that to happen. Okay? So it's going from areas of high concentration to an area of low concentration. Now, we can increase the speed of diffusion. So there's several different ways to do that. Temperature, pressure, are both ways in which we could do this. Okay? So that's what I'm going to do as well. I'm going to speed up the molecules just a little bit.
Oh yeah.
And then you can see that it's spread evenly throughout this container. It's not separating, so we know that this mixture has diffused evenly.
Love it.
That's the same way with this example here as well. So again, over time, that's what you should see. You should see a homogeneous mixture where it all appears the same throughout the entire container. So what is this called? This idea that we have an evenly distributed amount of molecules in a given area. That is called dynamic equilibrium. So that's what our cells want to do as well. Our cells want to be able to have the same amount of molecules both inside and outside of the cell. Okay, and so our cells will reach this dynamic equilibrium by diffusing materials in and out of the cell. And so it's going to continue to do that. So it's going to continue to go in and out of that cell in order to make sure to maintain that concentration.
Any spray that can clean up my knees.
Alrighty guys, our first question, okay? So what you're going to do is, you're going to draw this membrane, you're going to draw arrows to show movement. And then you're going to predict what would happen over time for this cell to reach equilibrium. Okay? So draw the membrane. So this is the membrane here. You're going to draw arrows to show movement and what would happen over time. Okay, so let's have Miss Becker's room. Okay, so Miss Becker's students, why don't you go ahead and provide the answer in chat?
I need a towel.
All right, again, Mrs. Becker students, I know you guys are out there. Go ahead and put your description of what would happen over time for the cell to reach equilibrium there in the live chat. Let us know what you're thinking. All right, here we go. We're getting a couple of answers coming in here from Mrs. Becker's students. So we have 3 of the particles to move over to the other side. About 3 of the molecules would move to the other side to create equilibrium. 3 of the molecules would move over to the other side. 3 would move to the other side. 3 circles would move to the other side. 3 would be on each side.
Awesome, guys. Those are some great responses, and exactly that's what's going to happen. So we're going to have 3 of these molecules moving over to the other side. So now remember that once they move, they're not going to be there anymore. Okay? So just to make sure that we understand that, I know that's pretty obvious, but we just want to point that out, is that they won't be on that side, they will be on that side instead. So good job on that, guys. All right, question 2. Very, very similar type of question. I'm going to go ahead and erase this here. So you're going to draw this membrane, drawing arrows to the movement, what would happen over time for this cell to reach equilibrium. Let's have my class answer this question now. Okay? So Mr. Wynn's class, predict what would happen over time for this cell to reach equilibrium.
All right, Mr. Wen's class. Let's see those answers there for this question number 2. Put your description of what you think might happen over time for this cell to reach equilibrium. Let us see what you got. All right. We got them coming in fast now. We have 2 circles would move to the other side, 2 would move. 2 would move on to the other side. 2 of the circles would move to the other side, 2 would move. 2 of the circles would move over to the other side.
Alrighty guys, so here's what we have. So we have this molecule here. This molecule will move over this side. This molecule will move over this side. Boom, we have dynamic equilibrium occurring.
Whoa, Mr. Wen. Can't do that. Doesn't work that way.
Well, why not?
Well, what we have to look at, I mean, this circle right here, how's it going to fit through that little old hole?
Are you saying that this is too big?
I'm saying that's too big to just move across the membrane to the other side.
Oh, okay. So we can't move over to the other side, and instead it is currently at equilibrium with these particular molecules, or it needs something else. Okay? And so in this case, none of them will actually move over to the other side based off of this setup here.
A little bit of a trick question there, Mr. Wen.
That's why I gave it to my students.
There you go.
All right, so diffusion in a cell. So this is exactly what we just exhibited. And remember, again, this cell membrane is fluid. It's moving. It's wanting to make sure that it's able to accommodate the different environments. And so in terms of the smaller molecules, they're able to diffuse across. They're able to go through this cell membrane. These are things like oxygen and CO2 they're able to go through. However, we have these larger molecules that aren't able to go through because, again, they cannot fit through the cell membrane. And so we want to make sure that we have a way in which we're able to get those large molecules across, because our cells still need amino acids. They still need carbohydrates, and they still need lipids. That method of getting larger substances across is called facilitated diffusion. So that facilitated diffusion is the movement of materials across the plasma membrane using proteins. That's what I want you to underline, star, circle, using proteins. Okay, so that's a really, really important distinction to make with facilitated diffusion. Alrighty, so we have these membrane proteins and these membrane proteins as well. And by the way, it says channel protein and carrier proteins. Those are specialized types of membrane proteins. So in terms of these membrane proteins, they act as a bridge to allow those substances across. Okay, so think of it like a bridge and making sure that these larger substances that normally would not be able to go through can go through. All right, I'm going to ask you an important question. So give me two differences between facilitated diffusion and simple diffusion. And then also give me two similarities between facilitated diffusion and simple diffusion. Now, in the chat, I'm only going to ask you for one of each, okay? So Mr. Ewald's class,
Yeah.
can you give me one similarity between facilitated diffusion and simple diffusion? Can you give me one difference between facilitated diffusion and simple diffusion?
All right, Mr. Ewald's class, I know you guys are out there. In that live chat, why don't you go ahead and throw out one similarity and one difference between facilitated diffusion and simple diffusion. You guys are rock stars. So I know that you will be able to provide these good answers. All right, keep those answers coming. We got a couple that are coming in. So far, we have a difference is that one of them must use proteins to transport. And it looks like we have a similarity. It goes from an area of high concentration to an area of low concentration. All right, another similarity. Both are movement of materials through the cell membrane.
Absolutely, yeah.
All right, keep those coming. Let's see if we can get a couple more before we have Mr. Wen move on.
Alrighty, so again, the important thing is to make sure to have these in your notes and also something that you're going to submit later on. So, two differences between facilitated diffusion and simple diffusion. One of those differences is absolutely what you guys nailed. Okay, one of them uses the membrane protein while the other one does not. So facilitated diffusion uses membrane proteins, simple diffusion does not. Another difference is the size of the particles. So facilitated diffusion allows for larger substances to move across the cell membrane, whereas simple diffusion can allow for smaller substances to move across the membrane. Now, two similarities that you guys had, absolutely 100%. The similarity between facilitated diffusion and simple diffusion is the diffusion of an area of higher concentration to an area of lower concentration. Okay? Another similarity that we see is that neither one of them uses energy. Okay? Neither one of them uses ATP. So we want to make sure that, again, that it's important to understand that there are similarities and there are differences between facilitated diffusion and simple diffusion. If it helps, facilitate means to help. Okay? So if you can remember that facilitated means to help, well, what's helping the diffusion? The membrane protein. Alrighty, so another question here. Drawing this membrane, drawing arrows to show movement. I'll predict what would happen over time for this cell to reach equilibrium. Okay, so again, very, very similar to what we saw, but now all of a sudden, we have this structure here. Okay? Make sure to label this structure and what this structure is, because that's important. Alrighty, now, remember the previous question, the holes or the gaps in that cell membrane were too small. So, if you were to do this, that would not work. Again, because the size of those molecules are too large for the pores or the holes in the membrane, it has to go through this membrane protein. So whenever it goes through that membrane protein, you're absolutely right. It's going to go from an area of higher concentration to an area of lower concentration. And you're going to have two that go across.
Bye, Brooks. Bye, Ramsey. Bye,
Love it, Mr.
Emily.
Wayne.
Bye-bye. Bye-bye, Jennifer. That was fun.
Okay, so the last type of passive transport we're going to talk about is osmosis. So osmosis is special because it uses this word water. Okay? So again, start, circle it, underline it, whatever the case may be. It is the diffusion of specifically water across a selectively permeable membrane. And again, remember that selectively permeable means that we're able to control. We're able to filter things that we need and things that we don't need. So we have three types of solutions. We have an isotonic solution, a hypotonic, and a hypertonic solution. Now, I don't want you to get all confused or think that these words are way too complicated. Okay? So what we're going to do is we're going to break the words down in order to make sure that it's easier to understand what's going on. So for an isotonic solution, and again, we're focusing on these prefixes here. Okay? So we're focusing on those prefixes. So in an isotonic solution, the prefix iso is very similar to what we have for words like isomer or isometric. And that means same. An isometric exercise is an exercise that works the same muscle groups. An isomer is molecules that have the same chemical formula. In terms of hypotonic, that prefix hypo, you might have heard of a hypodermic needle, or you might have heard of hypothermia. So again, all of those refer to the word below. So in the case of a hypodermic needle, it goes below the epidermis. So in terms of hypothermia, that's where your average body temperature goes way below than what it should, almost to the point where it's critical. And then hypertonic, so the word hyper is what we see in terms of hyperactive individuals. Okay, you know who you are. Okay, but in terms of hyperactive, my daughter is hyperactive, by the way. And that means that their activity levels are way above their peers, or way above what should be normal. Okay, but in terms of hypertonic, so we're going to talk about all these different words and how they relate to osmosis and the diffusion of water. Okay, so in an isotonic solution, water and dissolved substances diffuse into and out of the cell at the same rate, which means that we have water that goes both in and out of this cell.
So people just figure it out.
The reason why we have this same rate is because the concentrations of inside the cell and outside the cell are relatively the same. And so it wants to make sure to maintain that dynamic equilibrium.
Ellie, I can figure out by myself.
Oh.
We heard you. I collect little flowers
In a hypotonic solution, so hypotonic solute concentration is higher inside the cell. So water will diffuse into the cell. Now, there is a very, very easy way to remember this. Water will always go wherever there is more solute. Okay? So, in the case of this example, there are more of these solute molecules, these orange dots, than there are on the outside. So water will want to go into the cell, and it causes that cell to expand.
But I'll never show my stomach. Or it'll look weird. See, I'm nice one.
Okay, and then finally, the last one is going to be your hypertonic solution. So in your hypertonic solution, the solute concentration is higher outside the cell. So water will diffuse out of the cell. And again, remember the general rule. Water will like this. If you're out in the ocean, you're dying of thirst, you start thinking, you know what? Drinking ocean water is a great idea. Mr. Ewald, why is drinking ocean water not a good idea?
Well, when we think about ocean water, we know that ocean water has a very high concentration of salt. It's salt water. So if you go into the ocean and you're stranded out there and you start drinking water, you are bathing your cells in water that has an incredibly high concentration of dissolved salts, right? And you just said that water wants to move towards the higher concentration of solutes. So it's going to move out of your cells. No bueno.
Yeah, don't do that, okay? So again, as desperate as you might want to drink that water, you definitely should not drink any salt water.
Words of advice to live by, right there. Love it.
Alrighty, so question 5, draw the picture, draw arrows to predict the movement of water. So in this case, we have, and here's what I want you to focus on, these percentages. Solute, 75%, water, 25%, solute, 45%, water, 55%. And is this going to be a hypertonic, isotonic, or hypotonic, isotonic solution? And what I would like you to do is to make sure to draw the arrows to predict the movement of water. Alrighty, so in the chat, here's what I want. Okay, so let's have anybody from Mr. Ewald, Ms. Becker's, or Mr. Wynn's class that's last name is A through O. I want you to go ahead and put in the answer and tell me, where is the water going to go? Is it going to go into the cell? Is it going to go out of the cell? And then what type of solution is this? Is this a hypertonic solution, a hypotonic solution, or an isotonic solution?
So all students, Mr. Wen, Mr. Ewald, Mrs. Becker, last name A through O, throw those answers down there in the live chat for your prediction of where the water will go and what type of solution we would have or we have based on this picture. I know I have some A through O last names in my class, so I'm hoping to see you guys in the live chat.
Now that was awesome.
Me too. Me too.
There you go. All right, so we're starting to get a few answers here. We have hypertonic solution, hypertonic, hypertonic. It will move out of the cell. Water will still move outside of the cell. It's hypertonic. Water will move out of the cell.
Alrighty, good
Yes.
job, guys. And that's exactly what we want to make sure everybody understands. So we're going to focus again on that solute concentration. So this solute concentration being 75%. This solute concentration being 45%. So comparing those two, we know that there's more solute concentration on the outside. So water will move where there's more solute. Alrighty, same idea. Okay, so again, solute concentration being 40%, water being 60%. Inside the cell, 45%, 55%. Is this going to be a hypertonic, hypotonic, or isotonic solution? So go ahead and throw that in chat. Okay, so Mr. Ewald, Mrs. Becker, in my class, if your last names are P through Z, go ahead and throw your answer in chat.
P through Z. Great job on that last question, guys. Here we have all students' last names P through Z. All classes, all students P through Z. Throw those answers down there in the live chat. You can still see it. All right, let's see those answers. All right, P through Z, let's get those answers. Our first one, we did have a guess of isotonic, so we'll talk a little bit about that here in a second. The next one we had was hypotonic.
All right. Keep those answers coming.
Keep those answers coming. We had a hypotonic, water moves into the cell. We had another hypotonic. Miss Becker sending out the bat signal to see if Adam, Cecilia, or Brady are out there. Or the Becker signal.
I like it, the Becker signal.
I like it too. Hypotonic, 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 hypotonic.
Alrighty, so you guys are absolutely right that it is your hypotonic solution. So again, we are going to look at and compare the two solute concentrations. So the solute concentration outside the cell is 40%. The solute concentration inside the cell is going to be 45%. which means that there is more solute inside the cell than outside the cell. And what is that general rule again? Water will always go wherever there's more solute. Now, if you had an isotonic solution, that means that the solute concentration would be the same inside and outside the cell. Okay? So, we're going to see that. So in this case, we have our solute concentration being 45% outside. We have our solute concentration of the cell inside that's 45% as well. So this is your example of your isotonic solution. Now here's what I want you to put in chat. What will the arrows look like? Okay? So what would the arrows like? So we know that this is an isotonic solution, but what would the arrows look like? So in chat, if you are a birthday that is between January to July. So January to July, I want you to go ahead and tell me what the arrows would do or what would happen with the movement of water.
January to July birthdays. Actually, that's me. Can I go ahead and answer that?
Well, we're gonna leave it to just students. I know.
Oh,
I'm
okay.
sorry, Mr. Ewald.
Man,
It would include me though. So,
I like it.
but I wouldn't be able to answer.
Correct. All right, so we got some coming in already. We got isotonic. One arrow pointing in and one arrow pointing out. Equal, pointing in and pointing out. We got another isotonic. The arrows would be going both ways. Isotonic, isotonic. One arrow in and one arrow out.
Nailed it, guys. That was.
Yes.
Ever seen that show, Nailed It? You guys did the opposite. Like, you did nail it, but you didn't nail it badly. Okay? You guys did a good job. All right, so in this case, there would be two arrows that go both in and out of the cell. Okay, so good job on that. It is an isotonic solution because of that 45%, 45%.
Love it, love it, love it,
All
guys.
right, so the last thing we're going to do is this live egg demonstration. So in this case, I want you to go ahead and copy this chart down. So what we have is we have a couple of eggs. We used to have 3. We don't talk about the third one, but we used to have 3. Now we have 2. So in this case, what we did was we removed the shell of the egg. The shell of the egg acts like a membrane because it prevents a lot of different things from getting to the albumin, the yolk, the actual egg itself. And so by removing that eggshell, now we can see a selectively permeable membrane. Okay? So we placed one of those without a shell in water, and we placed one without a shell in Karo syrup. And what that Karo syrup is if you had syrup on your pancakes before, it's like that. It's that particular consistency. And so we then masked it prior to putting it in, and we have 66.5, which is the initial mass, and 62.3 for the Karo syrup before we place it in these different solutions. We're going to show you what the eggs did after 24 hours of being placed in these solutions. It's really, really cool. Now, of course, you can try this at home as well. Okay, so take an egg with your parents' permission. Okay, so again, parents' permission is super duper important. Take an egg and place it in vinegar. Wait probably about 24 hours, and you're going to see that eggshell come right off. It's really, really cool.
All right, so I'm going to move you guys. Hello, everybody. I'm going to move you guys over here so that we can see, there's Mr. Wen, so that we can see our setup that we have here with the different eggs. So we have a digital scale, right? We're going to use that to take the mass. We're going to measure that mass in grams. And then over here we have our 2 different eggs. So the first egg that Mr. Wen has there, just check that bad boy out, right?
You can see the yolk too, which is super cool. See that yolk that's just sitting inside there?
Awesome.
And so we're going to place this on the scale.
So, Mr. Wen, which solution was this egg in?
So this was the water solution. So it's been sitting in water for 24 hours. And so the initial mass was 66.5. So what happened? Well, it obviously gained mass. Okay, so that the final mass is going to be 88.38.
88.38 grams. And here's my favorite egg from this demonstration, from this experiment. So check this bad boy out. You got it.
How do we even pick it up?
Got it very gently.
Oh, very gently. So notice that it is literally, I can touch the yolk. That is super cool. Okay, and it is nice and slimy and grimy, and you can even see the folds of the outer membrane.
Oh yeah.
Okay, we're going to place this on the scale.
And just a quick refresher, this egg was placed in what solution?
This was placed in the Karo syrup.
So this was placed in the Karo syrup. And here is our final mass. After 24 hours of being in Karo syrup, the egg now has a mass of 35.01 grams. It started off at 62.3 grams. And now our final mass is 35.01 grams. How cool is that? Just dissolving the shell of the egg using vinegar and then placing it in some different solutions. Get you guys straight again.
All right, so do me a favor, okay? So on this one here, make a third column over here and tell me what type of solution that is. Okay, so if you need a reminder, is it a hypertonic solution? Is it a hypotonic solution? Is it an isotonic solution? Okay, so again, that third column there, solution type, and then you're going to tell me hyper, hypo, or iso. Alrighty guys, that is it for the live lecture. Thank you guys so much. I know I had a blast. I know that Mr. Ewald had a blast.
Absolutely.
And so again, it's always nice to be able to talk to you guys, even though it's virtually. Okay, so in terms of any other questions, make sure to turn in the questions by the end of your class period. Check your Schoology for instructions on how to submit it. Check Schoology to see whether your teacher has assigned any more work. I know for my students I've assigned more work, so you want to make sure to check that. Mr. Ewald, what about you?
So for my class, we should have a discussion that is posted on Schoology in our Unit 3 materials. All right, so jump in there and go ahead and answer those questions for the live lecture. We do not just yet have any extra work. We're going to hold that off until we do our next live lecture. So for you guys, for Mr. Ewald's class, your main goal is to go ahead and finish that discussion before the end of the day today. All right, awesome stuff.
All right, and then our next time-bound event will be a live lecture again on active transport this time. Alrighty guys, it was good metaphorically seeing you guys, and I will see you next time.
Yeah, missed you guys. Thanks for joining. Mrs. Becker, Mr. Ewald, Mr. Wen's class, thank you so much for joining. We'll see you guys very soon. All right, adios.